the structure and function of organelles. The objectives I would like to achieve: learn the names of each organelle within a cell, describe the structure and shape of each organelle, describe the function of each organelle. I want to start by focusing on the nucleus. Let's look at the structure of the nucleus first. It is that rounded portion in the center, right? This is the nucleus surrounded by the nuclear envelope. The folded organelles off to the sides are actually part of the endoplasmic reticulum. We'll be focusing on that later. For now, focus on this rounded bit in the middle with all the squiggly DNA inside of it. The nucleus consists of a nuclear membrane filled with pores covered with holes and is filled with the nucleoplasm. The nucleoplasm is a matrix in which the materials within the nucleus is suspended in. This acts simultaneously as a protection system as well as a medium that can be moved through. So this is important because you want to have a medium for all the working parts to move through. Otherwise, it will have to function without a gel-like substance. So it would have to walk on the outside of that nuclear envelope, or everything would be laying flat on the bottom. And if you move that cell around, it would slam parts into each other, and this could cause damage. So having that nucleoplasm really softens the movement and makes sure that nothing damages each other when they run into each other. So within the nucleus, genetic information is stored, replicated, and transcribed. A major component of the nucleus is the transcription factor, making copies of DNA segments into messenger RNA, mRNA. This messenger RNA, this mRNA, is transported through the nuclear envelope using those pores to then be translated to proteins. This process is required to make all the functioning parts of a cell as well as maintaining their functions. So all those parts within a cell come from the blueprint, that is the genetic information from DNA, right? From the DNA. Without the nucleus, the DNA would be spread throughout the entire cell, making transcription and translation a process that would require much more time and effort to complete. If everything is diffused through the cell, you would have to wait for an enzyme to get across the cell to take care of a certain component, or a protein to travel all the way across the cell to read a portion of DNA. By keeping the contents of the nucleus more closely bound via the nuclear envelope, it ensures less work and less luck. So all of this mean that less work is required to complete the process, just to live, right? So that's what we're looking for, is more efficiency. Now, if you dive deeper into the nucleus, you'll find what's called the nucleoli, or the nucleolus, and that will be what we talk about next. So let's now dive into the nucleoli. Okay, the nucleoli is found within the heart of the nucleus, making up about 25% of its core. This is a membraneless area, so when you dive into the nucleus, you do not go through a membrane and suddenly realize you're in the nucleoli. Instead, it is rich with RNA. This RNA, a lot of it is ribosomal RNA, or rRNA, okay? Because the nucleoli is responsible for processing and transcribing rRNA. So with all of this genetic material required to synthesize ribosomes, subunits of ribosomes are synthesized within the nucleoli. In other words, all this genetic information for ribosomes are there, so they're making parts, the subunits, the parts of ribosomes, so they can form ribosomes. This furthers the point that the nucleus facilitates protein synthesis. You have DNA, which goes on to be formed into protein, as well as ribosomal subunits that are formed within the nucleus in order to make ribosomes, whose whole process is to make protein. Recent studies have shown a connection between the nucleoli and certain cancers. It has been found that size, shape, and activity levels can be a predictive tool to diagnose cancers early on. If a nucleoli process of ribosomal biogenesis is overproducing ribosomal subunits, it can be a precursor to cancer. You can in fact see these in a cell. If you look into a cell under a microscope, look at the nucleus, the nucleoli can actually show up as a darker spot within the nucleus if it is being active, or more active than a non-active nucleoli, which would show up as no darkening spot there. You very likely would not be able to see that darkening spot. From the nucleoli, we are now going to look at the rough endoplasmic reticulum. So the rough endoplasmic reticulum, or the RER, is connected directly to the nuclear envelope. It is contiguous with the nuclear envelope. It is so named rough due to the fact that the outer layer of the rough ER is covered in ribosomes. These ribosomes provide the RER with its function, synthesizing proteins. The mRNA, the messenger RNA that exits the nucleus after transcription, comes to the RER to be translated. This is where the information from the messenger RNA, the mRNA, is read. 3 nucleotides at a time, or a codon, to bind amino acids. When a sequence is read, that terminates the reading process. In other words, when the codons that say stop translating, stop adding amino acids, when that is read, the amino acids are then modified by the rough ER in a folding process, ensuring that these proteins fold correctly so that they function correctly. These proteins can then be delivered by the Golgi apparatus, which we'll talk about later, where it will be modified once more and packaged to be sent throughout the cell or out of the cell. The smooth endoplasmic reticulum, which as you can see here looks like an organelle with sacs inside of it. It's hollowed out, almost tubular. It is the smooth ER, and this differs from the rough ER because it's not covered in those ribosomes. It's not synthesizing proteins. Instead, it differs in how it functions, but it has some very similar functions as well. It all is based on what cell you are looking at. For example, across the board, the smooth ER are responsible for producing lipids. But if you were to look into certain endocrine glands, they are also responsible for producing steroid hormones that can be sent throughout the body. While if you look into liver cells, the smooth ER is responsible for metabolizing drugs or breaking down toxins. It helps filter the blood, as well as convert glycogen to glucose. Furthermore, in skeletal muscles, the smooth ER changes to a sarcoplasmic reticulum, which stores calcium ions. This storage is important for muscle contraction when stimulated by a nerve. So it's storing those calcium ions that are required in order to be able to flex or contract our muscles. The Golgi apparatus. As you can see from this image, it is folds of organelle. You can see that the organelle is made up of several folds. Okay, these flattened folds are known as cisternae. They can have between 4 and 8 cisternae within a common eukaryote. There are examples, however, of single-celled eukaryotes that have upwards of 60 cisternae. So a lot of variation in the numbers there. The grouping of cisternae are then segmented into 3 parts. You have the cis phase, which is closest to the endoplasmic reticulum. You have the medial, which sits between the cis and the trans phase. And lastly, you have the trans phase, which is farthest from the endoplasmic reticulum. Now you get some idea of what's going on here by the names. The fact that the cis phase is closest to the endoplasmic reticulum and the trans phase is farthest away tells you that things are going to be going all the way across this Golgi apparatus from the ER. Okay, it's going to be going through it, trans. The Golgi apparatus is responsible for taking in proteins and lipids provided by the SER and the RER via the cis phase. So everything's coming through that cis phase. It will modify and package these lipids and proteins to be delivered through the cell or sent out of the cell. The modification that occurs complete the process of turning the lipids or proteins into fully fledged molecules. Enzymes are used to attach or detach saccharides, fatty acids, or phosphates. This process takes place in the lumen, where it will also be determined whether to deliver the package intracellularly or extracellularly, in the cell intracellularly or out of the cell extracellularly. Once modification is complete, the package will be sent out of the transphase to begin the delivery process. Now, in order for a package to know whether to go in or out of the cell, it actually uses a sugar marker, a saccharide, a sugar marker, to determine where it is going. So this process that the Golgi apparatus uses also has been shown to remove excess calcium 2 ions from the cell, ensuring that we don't experience toxicity from too much calcium from sources like milk. So the Golgi apparatus helps ensure that we do not poison ourselves every time we have a glass of milk. Now we are looking at the powerhouse of the cell, or the mitochondria. The reason that it's called the powerhouse of the cell is because it is responsible for synthesizing adenosine triphosphate, or ATP. ATP is a molecule that, because of how it is bonded, it provides a high level of energy when that bond is broken. Due to this, the molecule is used throughout many reactions in the cell to provide energy necessary to complete reactions. Mitochondria are double membrane organelles. You can see that within this image there is this double membrane action. That double membrane is evidence that there was an endosymbiotic relationship that formed long time ago. So early on in the formation of eukaryotic cells, a little bacterial type cell, very like a precursor to mitochondria, joined in and made a relationship to the eukaryotic cell. In turn, it provided energy. Right? The mitochondrion provided energy to that cell, and it expected protection, housing. So, endosymbiosis. You can also see this from chloroplasts. Chloroplasts later on joined up with a different eukaryotic cell. And the reason that we know this is because not every cell not every eukaryotic cell has the chloroplast, but even plant cells have mitochondria. So that shows a difference in timing. So if you look directly at the mitochondria, they also have their own DNA, and they reproduce asexually. Due to a lack of means to repair that DNA that they have, mutations are very common within the mitochondria. Mitochondria being strongly tied to apoptosis, that means that mutations can inhibit apoptosis, leading to cancer. Finally, the mitochondria is passed down from mother to child. due to the mitochondria being found in the egg, while the mitochondria in the sperm are often destroyed. This can lead to the child getting the mother's mitochondria. When it comes to the number of mitochondria found within a cell, it depends on the cells you are looking at. Blood cells, for example, lack a mitochondria, while muscle cells can have upwards of thousands of mitochondria. Centrioles are an organelle found within the cytoplasm of the cell. Looking at it, at the structure here, you can see that there are a lot of tubules involved with the centriole. They are responsible for making microtubules. These are used for several purposes. One purpose is to act as a skeletal structure for the cell, providing structural support. A second purpose of those microtubules is to help organize the cell during cell division, used specifically during anaphase to align the chromosome to move them to opposite ends of the cell prior to splitting into daughter cells. Lastly, the formation of cilia or flagellum for the cells. This process is known as ciliogenesis. This requires a mother and daughter centriole to be within proximity of each other to form the cilia or flagellum. The last organelle that I'm going to cover is the lysosome. Lysosomes form from the transphase of the Golgi apparatus, so they are an organelle that forms from another organelle. They then fuse with the vesicles formed from endocytosis to break down the contents within those vesicles. Endocytosis is the process from when a molecule outside of the cell is brought in by invaginating the molecule with a portion of the cell. The vesicle that forms from this binds to the lysosome that is filled with enzymes used to break down many molecules. Lysosomes also carry out a process known as autophagocytosis. This process utilizes those same enzymes to break down parts of cells that are malfunctioning or old. The molecules broken down by this process return many of the resources back to the cell to be recycled. Sometimes autophagocytosis can occur on cell parts that don't need to be broken down or excessively to any and all parts of the cell. When autophagocytosis goes through this malfunction, it has been tied to diseases like Parkinson's disease. If you would like to dive deeper on this information of organelles and cells, here are 3 resources. Khan Academy, which does a great job breaking down each individual part and really going into detail. The Amoeba Sisters, which take such a complicated concept such as organelles and trying to visualize what's going on such a micro scale, and they simplify it, help you understand. And then lastly, the Nucleus Medical Media is a great video that takes what's going on and provides the information as we understand it.